So we're finally starting to discuss actual molecular techniques, and the very first of our molecular methodology discussions is going to be nucleic acid extraction. Following the lecture, you should be able to compare and contrast organic and solid phase approaches for isolating nucleic acids, to list and describe the functions of the enzymes and reagents utilized to isolate nucleic acids, to describe gel-based spectrophotometric and fluorometric methods used to determine the quantity and quality of nucleic acid preparations, calculate the concentration and yield of DNA and RNA from a given nucleic acid preparation, calculate the quality of extracted DNA and RNA, and list the advantages and disadvantages of commercial extraction kits. So what is extraction? Extraction is when you release nucleic acids from a cell, and you're going to usually release these nucleic acids to use them for other methodologies. And you always want your nucleic acids to be free of contamination. So you just want that nucleic acid, whether it's DNA or RNA, you want it to be nice and clean. So you don't want to have any proteins in the mix, carbohydrates, lipids. If you're extracting DNA, you don't really want RNA in there, and if you're extracting RNA, you certainly don't want DNA, so other nucleic acids. So there's various techniques to remove these types of contamination. When we do extraction, usually you're going to use enzymes. So some enzymes that are used are proteinase K, which digests proteins, RNase, which is going to degrade RNA and remove any non-DNA nucleic acids, and DNase, which is going to degrade DNA and remove all non-RNA nucleic acids. So the first step in extraction is to lyse or break open the cellular material. And this is done using conditions that you're only going to damage the membrane of the cell, but not damage those nucleic acids, because you need to use those nucleic acids for various other purposes. The common sample types where you're going to extract your nucleic acids are nucleated cells, tissue samples, microorganisms. You could use bacteria, viruses, you name it. Any cell that contains nucleic acids are the cell types that you are going to extract from. So the common specimens that you may obtain in order to perform nucleic acid extraction would be blood, a buffy coat, bone marrow, a solid tissue sample, lavage fluids, any microorganism, bacteria, virus, fungus, as well as organelles such as mitochondria. And in parentheses, that is an estimate of the amount of DNA or nucleic acid that you can obtain from that specimen. Different specimens need to be prepared in order to extract the nucleic acids. So if you have a bone marrow or a peripheral blood specimen, you usually are going to have to do a step before you actually perform the extraction, and we're going to talk about these. So you may have to do a density gradient centrifugation or differential osmolysis, things like that, before you actually get the correct cells in order to extract from. Your tissues, you may have to digest the tissues, mince the tissues, break them up into small pieces. Plants, you might need to homogenize or break them up with glass beads. Your nucleated cells. You want to obtain, if it's a blood specimen, which is going to be a common specimen for nucleic acid extraction, you want to obtain the white blood cells. And in a whole blood specimen, you're going to have a whole mess of red blood cells in there that don't have a nucleus, hence they do not have any nucleic acid. So in order to obtain those white blood cells from the whole blood, you're either going to do differential density gradient centrifugation, where you're going to mix either your whole blood or your bone marrow specimen with saline and overlay that over Ficol. And differential lysis, you can lyse different sections of blood or bone marrow based on differences in the cellular osmotic fragility. So we mentioned Ficol to do differential gradient centrifugation. So what Ficol is, it's a highly branched sucrose polymer. It doesn't penetrate biological membranes, so it's really nice just to separate the different portions of your blood. So once you pour your whole blood over the Ficol, you're going to centrifuge it. Once you centrifuge it, the white blood cells are going to settle right on top of the Ficol layer. Your red blood cells are going to be underneath the Ficol layer, and the serum is going to be right on top of the white blood cells. So you can easily remove your white blood cell layer from this gradient, and then from the white blood cell only layer, you can then do your extraction techniques. So here's an example of Ficol gradient, where you're going to take your blood and place it over Ficol. Ficol is a very dense substance, so the blood can sit right on top of it. You're then going to centrifuge it, and your blood components are going to separate, and you can find the white blood cells directly on top of the Ficol layer. So what you would do is you would take a pipette, remove the plasma, maybe put it into another container for other uses, and then you take another pipette, sterile pipette, and pull those white blood cells off of the Ficol layer. You don't want to pull up too much of that Ficol layer. You want to try to get just white blood cells and just a little bit of Ficol. And then you can use the red blood cells for a different purpose, or discard everything after you get your white blood cell layer from that Ficol gradient. In differential lysis, you're going to take your whole blood or your bone marrow and you're going to put it in a hypotonic buffered solution, such as a water, because water is going to be hypotonic and will lyse cells. So the thing about putting whole blood or bone marrow in a hypotonic solution, the red blood cells are going to lyse first. So you can lyse your red blood cells and then centrifuge, and your white blood cells will not be lysed, and they'll be in the pellet. You can pull off the lysed red blood cells and other debris, and you'll be left with your white blood cells in the pellet at the bottom of the tube, and then you can perform extraction techniques on your white blood cells. Tissue samples, usually in a clinical lab, are embedded in paraffin. So what you would need to do is remove that paraffin so that you can get to the tissue. Once you have your tissue, you need to dissociate it so that you can get to the cells in order to extract your nucleic acids. So tissue is usually going to be ground, homogenized, or minced before you do your extraction methods. Microorganisms, you're going to usually use enzymes such as lysozyme, zymolase, lysostaphin that are going to help break open the cell wall, because the cell wall is very thick. You can also do grinding or mixing methods. It's common to take a bacterial suspension and mix sterile glass beads and then vortex it at high speed, and the glass beads will help break open the cell wall. You can also use chemicals, usually detergents such as SDS or sodium dodecyl sulfate, with a strong base such as sodium hydroxide, and that will also break open those cell walls. So nucleic acid isolation. Once you separate your cells that you want to extract the nucleic acid from, you can use various isolation methods. And you want to isolate just that nucleic acid, so you want it to be pure. So there's different ways you can isolate your nucleic acid. You can use organic solvents such as phenol and chloroform. These are very common organic solvents that are used to extract both DNA and RNA. So what the phenol and chloroform does, it dissolves hydrophobic contaminants. You can collect the cellular debris and it's going to remove proteins. So you end up with nice, clean, pure nucleic acids. And that's always your goal with nucleic acid extraction. You want that nucleic acid to be nice and clean and not have anything else that might be in there that's going to affect another protocol that you might use. So what do these organic solvents such as phenol and chloroform do? So phenol and chloroform is going to form a biphasic emulsion. So you're going to mix your white blood cells or your lysed bacteria with phenol and chloroform. You're then going to centrifuge. Centrifuging is going to put the hydrophobic components like lipids on the bottom. Your DNA is going to dissolve in the hydrophilic portion, which is in the top, the upper aqueous phase. Cellular debris is going to be a white precipitate that sits in between the aqueous phase on the top and the hydrophobic phase on the bottom. So what you want to do is collect just the very top aqueous phase without picking up that cell debris, that precipitate. Then you're going to do further steps once you collect that aqueous phase to obtain your nucleic acid. So once you get that upper aqueous layer into a clean tube, you can precipitate the DNA from that layer using salt and ethanol or isopropanol. So your DNA is going to precipitate out, and at that point it's no longer a liquid, it's going to be a solid, and you could centrifuge the tube, remove the supernatant, and you'll be left with your DNA precipitate as a pellet. So you added salt to precipitate that DNA. So you want to remove some of that salt because it can affect other protocols such as PCR and things like that. So you remove the salt by rinsing the pellet in 70% ethanol. You let that pellet air dry, and then you dissolve your DNA in a buffer. So usually in the lab, the buffer that's used is called TE, and TE is a mixture of 10 millimolar tris and 1 millimolar EDTA. You can also dissolve your DNA in water. However, water might contain some DNases or enzymes that could degrade your DNA. So a nice buffered solution like TE is commonly used. You can also just use tris without the EDTA because EDTA can inhibit some PCR reactions. And in the clinical lab, commonly we're going to isolate our nucleic acid to perform some type of amplification technique on it. So you don't want to have anything in there that might affect a protocol that you'll use down the road. So this is an example of your organic solvent layer, where you have your hydrophobic components on the very bottom, you have your cellular debris and proteins as a white precipitate at the interface, and the top is a very clear aqueous solution that contains your DNA. So in organic DNA isolation, you're going to take your cells, you're going to lyse those cells with sodium hydroxide and detergent such as SDS, break those cells open. You are going to then put them in a buffer with acid and a salt. You're then going to use organic solutions such as phenol and chloroform to extract your DNA from all of the other components. And then you need to precipitate that DNA out using your salt and ethanol so that you can pellet the DNA and get rid of everything else. And this is just another figure. This is actually from your textbook, figure 4.1, which is showing the same thing. You can also use inorganic solvents to isolate DNA. And the reason inorganic solvents might be used is phenol and chloroform are toxic. So phenol, if you spill it on yourself, it'll actually burn through your skin. And chloroform won't burn your skin, but it is a type of a mutagen, so you really don't want to get it into your system. So a lot of people don't want to use phenol and chloroform in the laboratory because they are toxic substances. So you can use inorganic DNA isolation methods where you have your cells, you're going to lyse your cells with TE and detergent, your SDS, and you're going to precipitate out your proteins using sodium acetate. You want to remove those proteins so that you can then isolate your DNA using isopropanol. There are also solid phase methods, and usually these are commercially available DNA extraction kits. There's many companies, Promega, Qiagen, numerous companies that sell these extraction kits. Most of these kits use what's called spin columns, these little tiny tubes where you put your lysed cellular material into the column, and the column contains a high salt buffer. The DNA is going to adsorb to the solid matrix in the column. You're going to wash that column, get rid of all the debris, and then you're going to elute the DNA off of that solid matrix into a solution so that you have nice clean DNA. You can also have spin columns that use silica or beads, where your DNA is going to attach to the beads, and then you could elute the DNA off of the beads. So it's a similar method to the solid phase spin columns, but it uses beads. So for your solid phase DNA isolation, you have your cells. Again, these are kits, so they supply what's called usually lysis buffer. You're going to add that lysis buffer. It's going to break those cells open. You're going to add your second reagent, which is an acidification step. You're then going to put this mixture over your spin column. Your DNA is going to adsorb to the solid membrane at the bottom of the spin column. You're going to wash everything through that solid membrane. Only the DNA is going to stick. And then you use your elution buffer, which is a low salt and usually some sort of a TE, and you're going to elute your DNA out of that solid matrix into the bottom of the tube. So these are two examples of a Qiagen kit. They have a little spin column kit, and they come with reagent 1, reagent 2, reagent 3, and you just add your reagents to your lysed cells, put them into the spin column, centrifuge your spin column. Everything but the DNA is going to go through the solid matrix. You're going to wash the DNA, and then you're going to elute the DNA into a clean, sterile microcentrifuge tube. So the advantages and disadvantages of using these DNA or RNA, you can buy either extraction kits. So the advantages are that they don't use those toxic organic solvents such as phenol or chloroform. They're much more rapid. You can do an extraction in about 20 minutes, whereas when you do phenol chloroform extraction, it takes a couple of hours. You can easily estimate your DNA concentration based on the amount of cells you use before you actually start the extraction. The disadvantages of extraction kits are that they are more costly. You can buy a bottle of phenol chloroform, some SDS, some tris EDTA, and you can do 1,000 DNA extractions with these bottles. Whereas your kits, one kit that maybe can do 50 extractions can cost upwards of, between $300 and $500. So they are costly. Sometimes you don't get quite as high of a yield of your nucleic acid with the kits, but the kits have been improved over the years, so you can almost get just as high of a yield with the kit as you can with some of the more manual methodology. DNA may not be as clean, but once again, the extraction kits have become very advanced, and you actually get nice clean DNA and at reasonably high yields. So the major disadvantage with extraction kits are the cost. Other DNA extraction methods, let's say you have a limiting specimen such as fixed tissue, dried blood, bone, you may have to use other methodologies in order to obtain your nucleic acids. They actually do sell kits specifically for fixed tissues and dried blood and bone. Also, you could do rapid extraction methods so that you don't have to spend a lot of time with phenol chloroform or, spinning, centrifuging many different several times. So they do have rapid extraction methods. One thing you can use that's very rapid is what's called crude lysate. So what this is you lyse open your cells, and then that's it. Everything else in there, your nucleic acids have been released from the cell. So you can then you can lyse the cells, do a quick centrifugation, get that cellular debris in the pellet, and use the supernatant for an assay. The problem with using crude lysates is you're not cleaning that nucleic acid. You're just lysing the cells and using the material. So certain methodologies, a crude lysate, you're not going to have clean enough nucleic acid to be able to do certain methods. Crude lysates are commonly used when you're doing amplification methodology because it's very quick. You break open your cells using a detergent, you spin it down, you take your supernatant, and then you set up an amplification such as a polymerase chain reaction, and you have enough nucleic acid in there to get amplification. So you really wouldn't want to use a crude lysate to run on a gel to see your genomic DNA because it's just not clean enough, but it can be used if you do further amplification methods on the sample. So a rapid DNA extraction these rapid methodologies are commonly used before amplification methods such as PCR, because you just want to quickly get your DNA so that you can do your analysis on it. So you're going to take your cells, you're going to lyse them open, you're going to neutralize them, and you're going to set up a PCR. You can take any type of cells, whether it's blood, you can take a cheek swab, which would be your buccal cheek swab cells. You're going to lyse open those cells in some way. If it's a buccal cells, you're going to heat that specimen, and then you're going to do your amplification technique on it. You can also purify your DNA using what's called a Chelex resin. This is commonly used before you do PCR. So let's say you have tissue, such as hair roots. You would heat that tissue, you would mix it with the Chelex resin, and then you're going to centrifuge it. The DNA is going to be in the supernatant, the Chelex resin is going to be in the pellet, and that Chelex is going to remove the multivalent cations. So you end up with just that DNA in the supernatant. You would then use the supernatant for PCR. If you had fixed tissue, a tissue section from the histology lab, you need to get rid of the paraffin using xylene or ethanol, and then you need to digest that tissue using an enzyme such as proteinase K along with tris buffer before you're going to do some of the extraction methodologies. If you are doing amplification, you can just take the deparaffinized and digested tissue and then set up a PCR. So more of a crude lysate or rapid nucleic acid extraction. In order to obtain specifically mitochondrial DNA, you can do this one of two ways. One way is through centrifugation. So you can take your specimen, you can centrifuge at low speed, which is going to pellet your cells, your nuclei, your debris. Then you're going to take the supernatant and perform a high-speed centrifugation on it, and the mitochondrial DNA can be precipitated using ethanol. However, probably a more common way to analyze mitochondrial DNA is not actually to isolate the mitochondrial DNA. It would be to isolate your total DNA using all of the various extraction methodologies, and then using specific molecular methodologies such as hybridization or amplification that would be specific for your mitochondrial DNA. So you don't even have to take the extra step to isolate pure mitochondrial DNA in most cases. However, if you are in a research lab working on mitochondrial DNA, you will isolate the mitochondrial DNA from the total DNA. Methods to assess DNA, and we're going to talk about this in a little more detail in a few more slides, but you can use agarose gel electrophoresis. You're usually going to run your DNA with a known standard. You're going to use molecular markers, which have very distinct sizes, and you're going to see a nice, bright, distinct band on your agarose gel. Another way to assess DNA is using the spectrophotometer, and again, we are going to talk about this in more detail in a few slides. So let's talk about RNA. So the previous section concentrated more on DNA, so let's focus on RNA extraction. So the RNA extraction methodologies are very similar to DNA extraction. The main thing with RNA isolation is RNA is much more difficult to work with than DNA. RNA is very degradable. There are RNases everywhere. All around us are RNases. And so you're trying to isolate RNA and it's mixing with RNases that are everywhere, and before you can extract your RNA, it's already degraded. So there are specific procedures that must be followed if you are doing RNA isolation. Usually people working with RNA have a specific room, an RNA only room. And this room is RNase free, or as RNase free as it can be. So you can buy these solutions called RNase erase, and you could spray it to remove all the RNases from the environment. When you are working with RNA, you're always going to use gloves. You're always going to use RNase free tubes. You're going to use barrier pipettes, sterile pipettes. Everything has to be RNase free. You're going to buy special RNase free water. The kits that you would purchase for RNA extraction all are RNase free. So RNA, you have to just be a lot more careful when you're extracting RNA than when you're extracting DNA. Your specimen collection, either bone marrow or peripheral blood, usually using acid citrate dextrose, heparin tubes. There's specific RNA tubes that can be used. If it's a tissue specimen, you need to process it immediately or freeze it at minus 70 degrees in liquid nitrogen, or if it's a fixed embedded specimen, you have to do your protocols where you remove the embedding materials. So for bone marrow, again, you're going to do or for peripheral blood, your density gradient centrifugation or your differential osmolysis. For tissue, you are going to freeze, you're going to grind, you can, crush that tissue up. For bacteria, plants, and fungi that have thick cell walls, you can homogenize, vortex with beads, and use enzymatic digestion. Lab preparation. So we already said usually you're going to use a special room for RNA work. Your bench, everything has to be RNase free. You're going to wipe down with RNase away, RNase erase, RNase sap. There's many different RNase removing materials you can buy through any of the purchasing companies. Everything should be disposable and certified as RNase free. So you're going to want to purchase everything and keep it in a special area that's all RNA ready area. RNA isolation, you're going to use either organic methodologies, your phenol chloroform, or solid phase methodologies, and just like DNA, there are RNA extraction kits readily available. So for organic RNA isolation, you're going to lyse your cells using guanidine. You're going to extract using phenol and chloroform, just like with DNA, and precipitate using your ethanol. So very similar to DNA. Solid phase RNA isolation, these are commercially available kits. You're going to be supplied a lysis buffer to break open the cells. The RNA is going to be absorbed to the solid surface with a low pH. You're going to wash your RNA and you're going to elute the RNA from the solid surface using a low salt buffer. You can extract RNA from fixed tissue sections. You want to get rid of the paraffin using xylene and ethanol, digest your tissue with proteinase K, and then you're going to want to do an organic RNA extraction using phenol and chloroform and precipitate using your ethanol. Remember, there are various types of RNA. You have messenger RNA, ribosomal RNA, transfer RNA, heteronuclear RNA, small nuclear RNA, double-stranded RNA, if you're extracting from a double-stranded RNA virus, or if your RNA has annealed to itself to make its specific secondary structures, as well as small and microRNAs. Let's say you wanted to look at just messenger RNA, because messenger RNA is going to tell you if a certain gene is expressed or not. So if you are isolating total RNA, 80 to 90% of that total RNA is ribosomal RNA. Your messenger RNA is only 2.5 to 5% of the yield of that total RNA extraction. So it's a much smaller amount of RNA for messenger RNA. So usually you're just going to isolate total RNA, and then you're going to do other methodologies like reverse transcriptase PCR or real-time PCR to look at your messenger RNA and not actually isolate the messenger RNA. However, if you did need to isolate specifically the messenger RNA, you can use oligomers, which the shorter term for that is oligos. An oligo is just a short piece of nucleic acid that you can purchase. There's companies that you can design oligos, or what's called primers, and they make them for you and send them to you. So these little oligos or primers, you can make them that are all T's or all U's, thymines or uracils, and these are going to be immobilized to a matrix, column, or to beads. So then you'll have your messenger RNA, and remember your messenger RNA has that poly A tail. So that poly A tail on the messenger RNA is going to bind to the string of thymines or the string of uracil oligos that's on that resin or bead. You can then wash everything else away and elute your messenger RNA off of the column or beads with a warm low salt solution. So your methods to assess RNA are the same as assessment of DNA. So you're going to use your gel electrophoresis, and you could see your RNA on a gel just like you could see your DNA on a gel. You could use your spectrophotometry, and for RNA you can also use fluorometry, and there's specific dyes that are used in the fluorometry method. So now we're going to talk a little bit more about these methods to look at the quantity and the quality of your nucleic acids, whether it's DNA or RNA. So in electrophoresis, the other lecture in this module talks much more in detail on electrophoresis. So this is one of the modules where you might want to listen to both of the lectures one after the other, because they kind of go hand in hand. We're talking a little bit about agarose gel electrophoresis in this lecture, but we're not getting into the details that we get into in the second lecture here. So electrophoresis using either agarose or polyacrylamide gels with your intercalating DNA or nucleic acid agents such as ethidium bromide. Spectrophotometry, where you're using a spectrophotometer to look at light transmittance. So your nucleic acids absorb light at 260 nanometers nanometers through adenine residues. Your proteins absorb light at 280 nanometers through tryptophan residues. And your fluorometry is a fluorescent spectroscopy, and it's going to measure the fluorescence related to the the nucleic acid, I should say, concentration. So for electrophoresis, you're going to resolve a portion of that nucleic acid that you just extracted. Usually you're going to make a dilution and take a small amount, maybe 5 microliters, and run it on an agarose gel. And you can do a rough quantification by comparing the intensity of the band with those of the marker. And they do have specific quantity markers that you can use. You can also look at the quality of the DNA run on the agarose gel. And we're going to see some pictures of these. So how the nucleic acid looks on a gel depends on the type of nucleic acid that you've extracted. So plasmid DNA, and we're going to talk a lot more about plasmids when we talk about recombinant DNA methodologies in a couple of lectures. So plasmids are circular, self-replicating pieces of DNA, and many bacteria have plasmids in them. And plasmids are commonly used in the laboratory for recombinant DNA technologies. So plasmid DNA, if you were running a plasmid on a gel, you're going to see a nice, bright, single supercoiled band that can move through the gel quite readily, so moderate mobility. Genomic DNA. Now, genomic DNA, the DNA that's coming from those cells, is a huge piece. It's a high molecular weight. So you're also going to get a nice bright band, but it's going to be very low mobility. It's not going to be able to move through that gel because it's so huge. RNA, you're commonly going to see two very distinct bands, and those are your two bands of ribosomal RNA. So here is a picture of a gel, and you can see on the lanes that are labeled M, that is your marker. So you're going to run a molecular weight marker, and each band in your marker has a different size. So then you can compare your nucleic acid band with the location of the band in the marker, and you can estimate the size of that band. So lanes 1 through 5 are genomic DNA, and you can see nice, bright, large band that does not run very far through the gel, so low mobility. Lanes 6 through 10, you have total nucleic acid, so you have your nice, bright, high band, which is your genomic DNA, and then your two lower bands, which are your ribosomal RNA. Lanes 11 through 15 are your total RNA. However, when you run total RNA on a gel, the only thing you see are the 28S and 18S ribosomal RNA bands. You don't see the messenger RNA, you don't see the transfer RNA. Transfer RNA is too small, messenger RNA, there's just not enough of it in the total RNA to visualize usually on a gel. Here's another gel, so you can see at the very top would be your chromosomal or your genomic DNA, nice bright band, doesn't really move all that much. Sometimes genomic DNA is so large that it can smear a little bit through a gel. So genomic DNA can be a little tricky to see sometimes. Then we have supercoiled DNA, which is commonly your plasmids are going to be supercoiled. Supercoiled DNA, because it's nice and tightly tightly wound up, which is your supercoil, that's going to run really quickly through your gel. So those are going to be lower bands. And then your RNA is very low, and sometimes you don't see the two distinct bands, you just kind of see a cloud at the very bottom of the gel. So if you just did a nucleic acid extraction, a phenol chloroform, you're going to get DNA and RNA and possibly any plasmids that are in there. In order to separate your DNA or your RNA, usually if you wanted DNA, you're going to use RNase to remove the RNA. If you wanted RNA, you're going to use DNase. If you want just plasmids, you do what's called the plasmid prep, which is one of those extraction kits specifically for plasmids, so that you don't get the genomic DNA, you only get the plasmid DNA. So if you just did a typical organic nucleic acid extraction and ran it on a gel, this could be what you see, because you could see the chromosomal DNA. Let's say it's a bacteria that has plasmids. You could see your plasmid and you could see your RNA. Over on the left, you see your marker, and here it shows the actual sizes in kilobases. So you can see the 10.1 kilobase, 8.1, 7. So you can look at the sizes. So in lane 1, your chromosomal or genomic DNA is so high, it's so huge, it doesn't even line up with the marker. But your supercoiled DNA, you have in lane 1, a band that's a little bit higher than 3 kilobases. If you run RNA, again, it's going to be very low on the gel. And here is RNA. You can see nice bright 28S and 18S ribosomal RNA. And these are 3 different cell types: liver cell, Helen Lane or HeLa cells, and Cas7 cells. This is an example of genomic DNA run on an agarose gel. On the very left and the very right, you have your molecular weight markers. All of the odd numbered lanes, 1, 3, 5, is genomic DNA. You can see one nice bright band, and it doesn't run very far on the gel. The even numbered lanes, 2, 4, 6, 8, this is genomic DNA that has been digested. And if you digest or cut your genomic DNA is so large that if you cut it up into small pieces, what it's going to look like on an agarose gel is it's just going to look like one big smear. And this smear is fragments of various sizes of that genomic DNA all cut up, being run all the way down the gel. So if you didn't intend to digest your DNA, let's say you did a DNA extraction and you want nice, clean, pure DNA, and you run it on a gel and you see that smear, you wouldn't be able to use that DNA because it's degraded. However, for certain techniques, such as doing southern blotting, you want to digest your DNA. So we're going to be talking about restriction enzymes and digestion of DNA in the next couple of lectures. So we talked about plasmids, these circular pieces of self-replicating DNA. Bacteria, a lot of bacteria contain plasmids because commonly plasmids have genes that bacteria need, such as antibiotic resistance genes. So if you wanted to isolate plasmids from a bacterial specimen, you can do a plasmid prep. using an extraction kit. And then you can also run your plasmid DNA on an agarose gel just like you would DNA or RNA. You're also going to use a molecular weight marker so that you know the size of your plasmid. The thing about plasmids is they are supercoiled, so they will run very far on a gel. But plasmids commonly get nicked up. So sometimes when you run plasmid DNA, you're going to see multiple bands on a gel. So we have our schematic on the left and we have our actual plasmid DNA on a gel on the right. So on the left, SC stands for your supercoiled DNA. And supercoiled DNA, nice, tightly wound, runs very nicely through the gel, so it runs the furthest. N stands for nicked plasmid, and a nicked plasmid has single strand breaks in it, so it's not as tightly wound, it's broken, so it doesn't run as quickly through the gel. R stands for a relaxed plasmid, which has double strand breaks. So whenever that plasmid is broken, it's not going to be as tightly wound, it's not going to run through the gel very well. And L is a linearized plasmid. And so that's going to run kind of in between a supercoiled plasmid and a nicked or relaxed plasmid. So on our actual gel of a plasmid, we have a linearized plasmid, and on the right lane you could see a top band, which is the nicked or relaxed plasmid band, and our supercoiled band, which is going to run a little faster, and faster means it's running further down the gel than your linearized band. And we're going to talk a lot more about gels and mobility in the very next lecture. So here's the relative positions of plasmid on a tris acetate agarose gel. So again, you're going to have your nicked or relaxed plasmid, which is cut, that's going to run the slowest. Your linearized plasmid is going to run the next slowest. Covalently bonded, you may see, although you don't commonly see that on a gel, you're going to see usually your brightest, most intense band, and usually the furthest down that you see is supercoiled. And sometimes you could see a lower band that's a circular, single-stranded plasmid, but you don't always see that band as well. If you were to take your plasmid DNA and digest it or cut it with an enzyme, you would completely linearize it. So the only thing you would see on the gel is the linearized plasmid. You would no longer see the nicked or the supercoiled plasmid. So here's an example of plasmid DNA run on an actual gel. And on the left-hand side, M, that's your marker so you can determine your sizes. Your C is going to be your control, your plasmid. So on the left-hand side of this gel, you have the plasmid called NB1. And on the right-hand side, you have a plasmid called NB0. And so in the C lane, this is undigested plasmid DNA. In the next lane, you have plasmid that's cut with different enzymes. So it's going to cut at various locations. We're going to talk about restriction enzymes later. What's important to know here is if you cut your plasmid DNA, it's going to run higher than your supercoiled DNA. So it's going to run similarly to a linearized or a relaxed plasmid because it's been cut with an enzyme. Here again we have digested plasmid DNA. You have your marker, you have lysate. So remember we said crude lysate is a little dirtier. You're usually only going to use crude lysate to perform amplification techniques. If you were to take a crude lysate and run it on a gel, you're going to see it's very smeary because it's got a lot of other stuff, all that cellular debris in it. So you're not going to see a nice crisp band on the gel. If you took your crude lysate and just precipitated it to get some of your nucleic acid out, and you take that supernatant and run it, you're going to see again kind of a smeary, not quite as dirty looking. In the P lane, that's purified plasmid DNA, and you could see the two bands. You have a relaxed circle DNA, and you have your supercoiled DNA. In your DP, that's plasmid DNA after it's been digested with two different enzymes. And what you're going to end up with are the pieces of the plasmid DNA that are left after it's been cut with these enzymes. So the enzyme digestion and plasmids and all that is going to make a lot more sense when we talk about restriction enzymes and recombinant DNA technology. Now, spectrophotometry, that's commonly used to analyze the quantity and the quality of your nucleic acids. So we already said nucleic acids absorb light at 260 nanometers through your adenine residues. So what you would do is there's what's called your absorptivity constants that you need to use to determine your quantity of either DNA or RNA. So for DNA, that constant is 50, and for RNA, the constant is 40. So when you do your spectrophotometer reading, you're looking at optical density or absorption. So you're going to do your spectrophotometer reading at 260 nanometers, see what that reads, and then you're going to use the appropriate conversion factor to determine the amount of DNA or RNA that you have. So when you are running your nucleic acid through the spectrophotometer, usually you're going to dilute it, because if you don't dilute it and you have too much nucleic acid in there, you're not going to get an accurate reading. So you want to dilute it so that it's in the linear range of that spectrophotometer reading. And if you do dilute your nucleic acid, that dilution is included in your mathematical formula to quantify. We're going to do an example of DNA. So you've just extracted DNA using one of the methods that we've discussed. You dilute it 1 to 100. You run it in the spectrophotometer at 260 nanometers, and you get an absorbance reading of 0.2. So in order to get your concentration of DNA in micrograms per mil, you take your 0.2 reading at 260 times your DNA conversion factor, which is 50 micrograms per mil, times your dilution, in this instance it's 100, that calculates out to 1,000 micrograms per mil. So that's the concentration of your DNA. You have 1,000 micrograms per mil. If you want to then calculate the yield, you have to take into account the total volume that your extraction was done in. So if your DNA was eluted in 0.5 mils of tris buffer or water, remember you had 1,000 micrograms per mil. So you would take 1,000 micrograms per mil times 0.5 mils to get 500 micrograms, and that's your total DNA yield. Now here's an RNA example. So in this instance, we extracted our RNA, we dilute it 1 to 10. So we take a portion of that RNA. We don't dilute the entire amount. We have our extracted RNA or DNA. We take a portion of it out and we make a 1 to 10 dilution of a small portion, maybe 5 microliters, 10 microliters. We measure the absorbance at 260 and we get 0.5. So in order to determine the concentration of RNA in this example, you would take 0.5 times your RNA conversion, which is 40 micrograms per mil, times 10, our dilution, so you get 200 micrograms per mil. Then if you want to get your yield of your total RNA, you eluted that RNA in 0.2 mils of your buffer. So you take your concentration, 200 micrograms per mil, times your 0.2 mils, which is your total amount. So you have a total yield of 40 micrograms of RNA. Your spectrophotometer, in addition to measuring quantity, also measures quality. So your nucleic acids absorb light at 260 and your protein absorbs light at 280 nanometers through tryptophan residues. So the absorbance of nucleic acid at 260 nanometers should be 1.6 to 2 times more than the absorbance at 280 nanometers. So you take your ratio of the absorbance of 260 over 280. If it's less than 1.6, that means your nucleic acid is not pure. So there's contaminants in it. There's too much protein and other contaminants to use for most of your other methods that you're going to use your nucleic acids for. Fluorometry can be used to measure your nucleic acid concentration because you're using DNA specific dyes. And there's various dyes that can be used. There's a dye called Hochstein stain, and this is a dye that combines with adenine thymine base pairs in the minor groove of the double helix. So it's very specific for double-stranded DNA. You have to use a standard that has a similar GC content to the GC content in your sample. And then you could use this dye, run it on a fluorometer, and you can measure the fluorescence, which is going to be equivalent to the amount of DNA that you have. You can use other DNA-specific dyes such as Pico green or Ali green, and these have a brighter fluorescence than the Hochstein. This is an example of what the fluorometer would give you. You would do a standard curve, you would measure your sample, and you're going to determine the amount of fluorescence, and you can then figure out the amount of DNA based on a standard curve. So things to keep in mind. The extraction method that you're going to use is going to depend on the cell type and what type of nucleic acid, DNA or RNA, you're trying to isolate, as well as the laboratory you're using. Also, the quantity and the quality of your nucleic acids is going to depend on the procedure that you use. So in summary, DNA is extracted by organic, inorganic, and solid phase methods. DNA can also be extracted using rapid methods or crude lysates. RNA extraction methods include both organic and solid phase methods, and DNA and RNA concentration, yield, and purity are assessed using agarose gels, spectrophotometry, or fluorometry.